Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. We are delighted to welcome you back to the third Aquatic Science International Conference 2025, organized by Faculty of Fisheries and Marine Science, Universitas Brawijaya, Indonesia.
Thank you to all participants for tuning back in on the second day of Third Aquatic Science International Conference 2025 under the theme Pollution, Protection and Progress, Navigating Water Sustainability in a Changing World. It is an honor for us to welcome back all the honorable speakers and respectable all invited guests and all participants.
Saya Dominic Antoine Rika Putri Sianipar
And I'm Muhammad Gavin Ahmad Wibowo
as the Master of Ceremony, and we would like to read today's agenda as follows. The first agenda is opening.
Second agenda is plenary session by keynote speakers and parallel session.
The third agenda is closing ceremony. Before we delve even further, let us open our agenda today by praying according to your belief and religion. Please, time is yours.
Thank you very much, dear audience. May the agenda run smoothly and successfully.
Excellencies, participants, ladies and gentlemen. Now we are entering the sixth plenary session of this event, which will be led by Dr. Citra Satria Utama Dewi, Master of Science. Dr. Citra is a senior lecturer specializing in ecobiology of marine organisms. To Dr. Citra, screen is yours.
Thank you very much for the master ceremony. Good morning, distinguished guests, respected speaker, and all participants joining the morning session of the third Aquatic Science International Conference organized by the Faculty of Fisheries and Marine Science, Universitas Brawijaya. Well, my name is Citra Satria, and it is my pleasure to serve as your moderator for this session. This morning session will feature a presentation from a respected researcher whose work focuses on deep sea microbiology and metagenomic, exploring life in one of the planet's most extreme environment. Our speaker for this session is Dr. Mo Han, MSc, PhD, from the Beijing Genomic Institute, BGI, China. Dr. Han currently serves as a senior engineer and leader of the Extreme Life Research Center at BGI Research, Sanya, where he studies microbial life in extreme environments, particularly deep sea ecosystem and their metabolic mechanism. He earned his PhD in molecular biomedicine from the University of Copenhagen, Denmark. And his work has been featured in top scientific journals such as Cell, Helion, and NPG Biofilms Microbiomes. Today, Dr. Han will present his recent study entitled SUMAT, a scalable metagenomic practice reveals deep sea microbial metabolic versatility and structural variation of bacterial VV hydrogenesis. Please join me in giving a warm welcome to Dr. Mohan. Dr. Mohan.
Hello,
Please.
Good morning, everyone.
Good
Thank
morning.
you for your warm welcome and introduction.
Okay, well, Dr. Mohan, screen is yours.
Okay, thank you. I would like to make sure everyone can see the screen of my slides.
Yeah, it's already.
Okay. Yeah. Okay, then I will start.
Okay.
So today my topic is about kind of a practice which may facilitate metagenomic studies. So why do we use metagenomics for our research? Basically, metagenomic sequencing may detect and quantify unculturable species, which is quite dominant in multiple kind of environments. And it can the results of metagenomic sequencing comparing to the kind of 16S RNA genes amplicon sequencing, it may provide a higher resolution in taxonomic annotation, kind of down to species or even strain level. And because of metagenomic sequencing, as its name, it sequenced almost all the DNA fragments and components in the sample. So it can also link functional potential with the taxonomic composition of a microbial community. And the last but most important, the cost of NGS, I mean, the next generation sequencing, is decreased during the last couple of decades. According to NIH survey, sequencing of a human genome, which is 30 gigabase pairs, can be done to less than $1,000 in United States, but it can be even lower. I mean, in China, we can do it with about $100. And as we can read from the top journals, metagenomic sequencing is a very widely used approach for studies in environmental microbial communities such as deep sea or kind of glacier or soil or kind of rivers. So it is very useful, but still metagenomic sequencing remains some challenges. The first of it is in some of the environments, the biomass can be quite low. For example, for the trench sediments, because the environment is really harsh, there are very few of life can live in there and tolerant the extreme environments, which means the biomass can be really low. For most of the samples we sequenced from the Mariana Trench, the DNA yield is less than 10 nanograms per gram of sediment sample. This is a very low value for the conventional sequencing approach. And the second thing is that the throughput of the traditional workflow, especially the wet lab part, is really low and label heavy, which also limited the studies, especially in the way of scale. Because when you try to conduct a large scale study, the sample size can be up to couple of hundreds or even which we did in the MIR project, it's 1,400 something. But if we do it manually, it can take quite long time and very hard work. And also the reference database for understudied habitats is still lacking because we still don't really know what kind of species are living there. And we didn't sequence it very comprehensively and deeply. So we don't have a high quality reference database for this kind of studies. To solve these problems, or to say to fill these gaps, we designed a kind of systematic comparison and benchmark to validate if an automated wet lab approach can in some way help the metagenomic study in extreme environments. So you can see that we use 3 different kinds of samples. We used 2 different DNA extraction methods and 3 different library construction methods. Of course, we also have different inputs of DNA for the sequencing. So let's ignore the annoying details and go to the results. Yeah. According to our analysis, the DNA extraction throughput can be significantly improved by using the automated approach based on the liquid handler. And low DNA input library construction method, actually it works very well. Even if you only have 0.1 to 1 nanogram of DNA, you can still have a very high quality in the sequencing results. You can see that for the raw reads quality and the KMA richness, and also the total length of assembled contigs, the results of this kind of automated low input approach is quite comparable with conventional method, which usually use 100 nanogram or even 500 nanogram DNA. And also the N50 and length of the largest contig also remains at the similar level when we use this automated method. But the thing should be remembered is that the performance of a certain approach for metagenomic sequencing varies between habitats. You can see that for the cold seed samples, if you only use 0.1 nanogram of DNA, some of the measurements, or say the performance, will be relatively lower, although it also can support some of the study purpose. But if you do want to sequence the most major part of the diversity in this kind of complicated samples, then you will need at least 1 nanogram or even more DNA to provide a large enough capacity of the library. And also, for most of the metagenomic sequencing studies, we will need to profile the outcome using different, softwares or approaches. So we compared some very well widely used softwares, for example, Metafilin and Kraken, and in this analysis we used the SourceMesh as a reference, because SourceMesh didn't really annotate the reads or the context. It just take the KMA frequency into account. So this is a very good reference profile for this kind of comparison, and we can see that for the wide for the well-studied environments, there are some missing labels, but the right side one is the ocean water sample. And in middle, it is the trench sediment, and on the left side, is the coast seep sediment. We can see that for the well-studied sample as ocean water filter, different softwares provide almost the same results. But for the trench sediments, because the relevant study is really limited, we don't have high quality reference database for the taxonomic annotation. So in this certain case, Kraken2, of course, we also conduct Kraken2 adjustment. The Kraken2 based on Kramer frequency and signature provide very similar results comparing to the soil mesh results. So this result suggests that if our study is focused on an understudied environment, which lacking a high-quality reference database, Kraken may provide more comprehensive and solid results compared to metaphyllum and other reference-based or alignment-based approach. And actually, in recent years, more and more researchers apply the de novo approach in metagenomic studies. So we also analyzed different methods or different combinations for this kind of de novo study. You can see we compared 4 in our study. The first one is very regular conventional pipeline. And the second one, we only use one binner rather than a combination of 3. And for pipeline 3, we use another binner, which showed higher performance when you have multiple samples. And for the last pipeline we started, we removed the redundant context in advance to low down the computing consumption. You can see by comparing the results of different pipelines, we can see that the pipeline 3, or to say the group sample co-binning approach, can provide more unique metagenomic sample genomes, or say MAX. But still, if you don't have enough resources to support this kind of heavy computation, you can still use conventional pipeline as MetaRep. It also provides very high quality and very high number of MAG recovery. But yeah, I think maybe some of you have already been kind of lost, because we should agree that the analysis of metagenomic sequencing data is quite complicated. there are kind of profiling, annotating, and de novo assembly, and so on. So how may we facilitate the use of these kind of weapons? How do we help junior researchers to use this modern technology for their own scientific questions? So we tried to develop a toolkit which more easy to use. This toolkit is for the whole procedure analysis of standard metagenomic sequencing data. It is very convenient because in this toolkit we provide two different kind of interfaces. Aside from the annoying command line, we also provide web page based GUI, which is more easy to use in your own computer, and the high performance computer in your lab. And we containerized this toolkit to facilitate the deployment of the software. And also, the approach is quite flexible because we used some human readable options to as alternatives of the numbers and mean list parameters in the command line. You can simply choose your research proposed and have that feature and your IT resource. Then you can have parameters for all the pipelines. This method is quite scalable. Actually, we tried it in our study in the Mariana Trench, and validated it can handle up to 100 terabases of sequencing data, which is the largest scale up to now. Here is a architecture diagram of our toolkit. You can see the basement of the tool is data processing. We control the low quality sequencing data, and we applied a reference database. And then we can use this software to do profiling, sampling, McBinning, and also taxonomic annotation. After the basic analysis, we can use some different controllers. As I mentioned, we can use options to adjust the parameters of these analyzing procedure. And all of these are controlled by either a web page interface or conventional command line interface. Yeah, here is a screenshot of our software. You can see that on the left side, this is the web page interface, which is quite simple. You can choose the diversity of your sample, the novelty of your sample, and the resource you have, and what kind of results you want. Then you just apply the job and wait for maybe a couple of hours or a couple of days. You will have the results you want. Oh, sorry. Using this approach, we showcase the power of this kind of simple or say easy to use approach. We analyzed the data from code zip segments and trench segments and found that there are very significant habitat specificity in bacteroid depths. You can see that all the strains, strain level genome bins STGB, we recovered from the water from the same clays. And while all the strains recovered from cold seep sediments are all in the other clade. And in plot C, you can also find that the specificity or the preference is quite strong and significant because obviously if a species or a strain is highly abundant in one of the environments, then it is almost absent in the other two. And this kind of preference, not just in the taxonomic position, but also in the function, or to say metabolic potential. For example, in trench sediments, we can find an enrichment in fatty acid oxidation related genes. And for cold-seep sediments, there are more genes related to complex organic matters degradation. This is because that cold-seep sediments is a very anaerobic environment, which is not friendly, as you say, to fatty acid oxidation. So most of the species inhabit cold seep sediments will kind of shift their metabolic pathway from aerobic oxidation into fermentation to gain enough energy for their survival. So overall, as a brief conclusion, we try to use a standardized framework which can be applied to diverse habitats. Either it is well studied or is completely unknown. Either it is quite simple or it's extremely complicated. And we decided to join the open source community, so everyone can find the code, the Docker images on the global public database and repositories. We will also continually upgrading these the computational modules with systematic benchmarking, and we'll try to integrate some multi-omic capable modules and some AI-driven modules to provide more functions. So generally, we would like to continually to provide more function, which of course with comprehensive benchmarking and comparison to provide more easy use metagenomic sequencing solution to every students and researcher. Yeah, so I think I saved a little bit longer for the upcoming discussions. So wish everyone have a happy study and good luck in your research. Thank you so much.
Thank you. Thank you very much, Dr. Han, for that fascinating and insightful presentation. We now have about 15 minutes for question and discussion. If you would like to ask a question, please raise your hand and or type it in the chat box. Well, okay. Dr. Han, while we wait for the audience, I would like to ask one question first. Is it okay?
Yeah. You're very welcome.
Okay, Dr. Han, your study introduced the SUM80 toolkit as a standardized and scalable approach for the deep sea metagenomic research. From your perspective, how could this framework be applied or adapted for other marine environments, such as a coastal and estuarine ecosystem, where microbial diversity is also very complex?
Yeah, actually, I should say that we are very confident in the application of our approach summit. Yeah, S2MAT summit in the environments you just mentioned, because actually coastal sediments are the most complicated environments as to our knowledge. It is, I mean, even more complicated comparing to the coastal sediments or soils in farms. So our approach is well designed and tested in different kind of environments, and we believe that if you want to use it in other environments which we didn't test, it can also work. And we are very happy to see the results or some feedbacks from researchers who worked in different fields.
Yeah. Okay, it's very interesting. And yeah,
Okay.
from your presentation, you mentioned discovering a new structural variants of bacterial vivi hydrogenesis in deep sea microbes. Could you share what this finding might mean for future study on microbial energy metabolisms or even potential biotechnological uses, maybe?
Yeah, actually, because hydrogenesis is an enzyme which transferred water to hydrogen gas or in the opposite way. But in most of the anaerobic environments, it generates hydrogen. So we believe that hydrogen is very good and clean energy for future production and industry. So in this study, we found two major structural variants of FeFi hydrogenase in deep sea sediments, which means there is a very high possibility we can find even more different novel hydrogenase in this kind of extreme environments. And we believe that some of these enzymes will show their tolerance of oxygen, because most of the conventional hydrogenase is very sensitive to oxygen. This limits the industrial application of hydrogenase in the clean energy. So, because in some of the deep sea extreme environments, the pressure is quite high, and the oxidative pressure is also very high. Hydrogenase in that environments may have a higher tolerance in oxygen and will be more easy to use in an industrial scenario.
Yeah, okay. Is there any question from the audience? Okay, Miss Lutfi is already here. Okay, Miss Lutfi, screen is yours. Open your mic, please.
Good afternoon, Dr. Mohan. I'm
Good afternoon, Luffy.
interested in your research about the hadal zone or in the deep sea, and how does the composition of the microbial community in the deep sea, is there any difference from those in shallow marine environment? And in your opinion, in what ways can findings from the hadal microbial study can enhance our understanding of the limiting of the ocean life and the origin of the microbial evolution?
Okay, so actually in this study we didn't analyze very large number of samples. It is not similar with the article published on Cell. But still, we can see that the taxonomic composition in different ocean environments is very, very different. For the sample filters from the sea above to 1,000 meters down, most of the species are well studied. Unknown species. But if we down to more deeper part of the ocean, the situation significantly changed, because we found that more than 60, 100, or even 90, 100 of the species are unknown or say unreported in previous studies. And in coastal sediments, most of the species are anaerobic, but in tidal trenches, most of the species are aerobic. This is because the difference in environment factors. We also find some kind of enrichment of microplastics and heavy metals in the tidal trenches, and also found the genes, protein encoding genes related to the degradation of these kind of plastics and heavy metal contained organic matters are enriched in this environment. So in our point of view, we think human activity can somehow affect this, deepest part of the ocean, and changes in the microbiota of this environment will also feed back to the normal environments and our, human society. Maybe in the context of global climate change, we need to be more aware about the shift of ocean microbiota. Yeah, this is my opinion.
So we still have a lot of challenges to fulfill the deep sea study.
Yeah, and actually, as to I know, Indonesia is a very very big. I mean, a big country, and you have a very long coast, maybe even the longest coast all around the world. Yeah, so study and kind of research in the ocean environment is quite important and helpful for the protection of the environments in Indonesia and the resource development, of course. So yeah, maybe we can have further discussion and cooperation in this field.
Yes, using the collaboration, I think it will be easily to study about how the deep sea environment in Indonesia. I think even the sampling is it's even not easy.
It's not easy, yeah.
Yeah, but yeah, maybe it's a secret, but actually
Sure.
Brin also joined an expedition last year and have already collected very many of valuable samples. We can start maybe in small scale studies and to learn how to use, for example, metagenomic sequencing approach to solve our problems, to provide high quality results. Yeah, even a small scale study can be a good start.
Yeah, okay. Thank you very much, Ms.
Thank you.
Lutfi and Dr. Mohan. We go to the next audience. Ibu Umi Zakia is already raised her hand. Ibu Umi. Yeah, okay. Your microphone, please. Unmute first. Okay.
Thank you. Thank you, dear Chitra, for the matter. As the moderator, thank you very much for the opportunity. Nice to meet you, Dr. Mohan. Thank you very much
Nice to meet you.
for bringing such an advanced, powerful technology in molecular biology, which is probably still a little new to us here. Oh, sorry, I probably have a little bit problem with my camera. I do hope you still can listen to my questions, can hear my
Yeah.
questions. Yeah. I read about eDNA as well. So
Yeah.
if I may or allowed to ask the question about is it the metagenomic technology is a part of eDNA analysis? So then by sampling with environmental DNA, then we analyze more or continue with the metagenomic technology, or is that right? Or am I wrong?
No, actually, I think because eDNA analysis is kind of more, I should say, based on the gene amplicon sequencing rather than the I mean the whole metagenomic sequencing. But the sample can be shared. I mean, for one sample, we can extract DNA once, and the yielded DNA can be used for either eDNA analysis or metagenomic sequencing.
I see.
So we will not need two samples for each.
Yeah. Oh, okay. We can. Yeah. Oh, I understand now that we can use the same samples for
Yeah.
both analysis. And I read that this metagenomic also able to be applied, implemented in many kind of samples like soil, water, and anything that comes in the environments and left by organism. And what I'm curious is with the same samples, you know that by eDNA we can actually know the biodiversity of the organism in the samples, looking at their DNA left in the samples. I wonder for your research, you're saying that it's for actually a bacteria rather than organism. Is that right? Or actually metagenomic is also for organism as well, not only just bacteria or microorganism.
Yeah, it's a very good question. And I should say metagenomic sequencing will provide information for all the organisms in the environment, either they are prokaryotes or eukaryotes. Actually, in one of an unpublished article we are working on, we assembled a fungal genome from tidal sediments. And this is the first fungal genome recovered by metagenomic sequencing from Hadal trenches.
Yeah. I see. Yeah.
But we also sequenced, algae and kind of small animals even in our samples, especially in the water filters, because there are all the planktons.
Okay. Yeah. And probably last question for the sampling itself. Do you need a special facilities or apparatus to do the sampling? For instance, like we have to sieve the water or the sediment or any kind of samples, for instance, like a trunk of tree or something like that. Do you think you need special sampling apparatus for that? Or do you need a...
No, not at all. We use the conventional approach for the sampling, because on a vessel or the equipment you have is very limited.
Yeah. So I see.
It is you will need no special equipment or reagents for collecting samples for metagenomic sequencing.
And so it means that when you try to do the sampling for the hydrothermal vents in the bottom of the oceans, you need to go there and take some samples from or
actually I think you can also use some clamps,
I know.
Collecting samples from deep sea. We have big clamps which can grab some sediments and rocks from the bottom of the ocean.
I see. Yeah. Oh, then.
And of course, if you have a submarine, ROV or AUV, it can be much more better. But it is not mandatory.
Okay. Yeah. Okay. And for instance, like samples, do you need to treat it with special things like how long the sample can be observed or something like that, or it's unlimited time you can use the samples?
For most of the samples, if you freeze it on the -80, the sample can retain its microbial composition within a couple of years, I should say. I didn't test the samples which have been stored for more than 5 years. I mean, I didn't test it, but maybe it can still work.
Okay, maybe last question.
Okay.
Last question.
Okay,
You're
last
very
question.
Last question.
Last question from Ibu Umi.
Yeah. I read that when we found the DNA or the probably predicted organism, then we have to sort of match with the gene bank or something like that. So suppose there's none in the gene bank exists for this kind of DNA or organism. What should we do?
Okay, this is one thing I mentioned, but I didn't go to detail. In my presentation, I mentioned that there is a so-called de novo assembling approach for metagenomic sequencing. In this situation you just mentioned, actually we do meet the problem for the trench sediments, because I should say nearly 90% of the species we found there are unknown. They are not in the public database.
Yeah.
So for these kind of environments, we first we assemble all the reads into context, and then we cluster the context based on the similarity of their sequence signatures, as well as their co-occurrence signatures in different samples. We use this kind of information to cluster some of the contexts which we believe are obtained from the same species into a cluster named as metagenome assembled genome, MAG. And these clusters we get from the data can be further annotated based on the public database, even they are not in the database, because we can because when the quality of the maps are high enough, we can do phylogenetic analysis to infer which taxonomic clade should the MAG be assigned.
Okay.
So we use this way to solve the problem on these unknown species.
Yeah.
Okay.
It's little bit longer for non-species, yeah. Okay, Dr.
Thank
Mohan,
you.
thank you very much, Ibu Umi. Next,
Thank
Dr.
you.
Mohan, we have one question in the chat box, and also we have one audience already raised his hand, but I'll read from the chat box first. Question from Vika from IPB University. NGS is expensive. She said like that. And the question is, how do I get around this regarding samples?
Okay, so I think
yeah.
there are two questions. One is NGS really expensive? I should say no, not really.
Yeah.
As I mentioned, at least in China, we can have 30 gigabase pairs of data with about $100. I think for most of the studies, the cost is quite affordable. We can also, combine some traditional, more cheaper approaches, such as the 16S RNA gene sequencing with metagenomic sequencing on some very important and essential samples. This can make the overall cost even lower. Yes, and So I don't know because it depends on your study design. I cannot simply tell you an overall guidance on the samples or say, but my idea is that if you are really short in cost in your budget, you can pick some of the most representative and important samples and subject to metagenomic sequencing. And for the rest of samples, you use a more cheaper and conventional way to analyze them.
Yeah, okay, thank you. Next question is come from Mr. Ekwan. Please, yeah. Screen is yours, Mr. Ekwan.
Thank you very much for Mrs. Moderator, Mrs. Citra. In this last question, I think we can focus to deep sea sampling. We know this extreme environment have so many maybe extreme compound like methane or other toxic compound. How you handle this problem? Because we know this compound can make a false result in DNA extraction or PCR or other method.
Yeah. Actually, you mentioned a very important issue in the experiments on this kind of extreme environmental samples, and my answer is a little bit maybe weird, because actually the solution is quite simple. You can dilute your DNA. Because as I mentioned in my presentation, for some modern approaches of library construction, the DNA input requirement is very low. We can use a very small amount of DNA to generate a very high quality of sequencing data, which means we can dilute the DNA as well as the contamination you mentioned. And by doing this, you can, in most of time, I mean, in our experience, you can increase the successability by about 10 or 20%, and you can have enough data from more than 80% of your samples. This is, I should not say good, but it is acceptable for most of the study designs. We can still lose maybe 10 or 20% of our samples, but the rest of data can still answer our questions. And also, you can run a gel. I mean, because electrophy is a very powerful weapon to remove contaminations from DNA compared to the the column purification. So if the sample size is not that big, if you have enough time to run some gels and recover the DNA from your gel, you can have a very high successful rate in your study.
Okay, it is very interesting. Mr. Ekwan, is it enough?
I think it's enough. Thank you, Prof. Mohan, and thank you for your joining to this conference. Thank you, Mrs. Citra.
Thank you for your invitation.
Okay. Well, thank you so much, Dr. Mohan, for that fascinating and eye-opening presentation. And thank you, everyone, for your active participation. To wrap up this session, We have learned how Dr. Han and his team developed SUMAT, a powerful metagenomic toolkit that makes studying deep sea microorganisms faster, more efficient, and more standardized. This work shows us that advances in technology are not just about the data, but about helping us to understand the beauty and complexity of life beneath the ocean surface and how much more there is to discover. On behalf of the Faculty of Fisheries and Marine Science, Universitas Brawijaya, and the organizing committee of the Third Aquatic Science International Conference, I would like to sincerely thanks to Dr. Mohan for sharing his inspiring research with us today. Thank you, Dr. Han, and of
Thank you
course,
so much.
thank you to all of you for being such an attentive audience. That concludes our morning session. I'll now hand the program back to our master ceremony. Thank you, everyone.
Thank you very much for the engaging discussion. We hope this meaningful exchange will serve as a stepping stone toward cleaner waters and a more sustainable future for all.
Moving on to the second agenda, ladies and gentlemen, we are going to begin our parallel presentation session. I would like to read the rules for the upcoming session as follows.
First, please rename your Zoom account, room number underscore name of presenter underscore institution. Second, please use the official virtual background provided by the committee on your Zoom screen.
Third, please join the breakout room according to scope assigned to you. Fourth, all participants will remain mute or in silent mode by the host, except for the resource person, moderator, and presenters when recognized by the moderator during the discussion.
Fifth, each presenter is allotted 10 minutes for presentation. A 5-minute for discussion or Q&A session will follow after 3 presentations or as scheduled by the moderator. 6, all questions may be submitted via chat using the format name underscore address to home underscore question or by clicking the raise hand icon to speak. The moderator will select and address questions in order.
Last, presenters who are not scheduled to present may join any room to wish to attend as observers.
For all presenters, you may check the distribution of respective room in the given book of abstract. Without further ado, we are going to begin our parallel presentation session shortly. Please enter your respective breakout room to begin the parallel presentation session.
Dear ladies and gentlemen, thank you very much to all the respectable participants for the outstanding contribution in our parallel presentation session. May this open the gates for further discussion and future collaborations.
Dear audience, before we move on to the next agenda, please kindly have a refreshment. We will continue our agenda starting at 1:15 p.m. in Western Indonesian time. Please be on time and see you.
Excellencies, distinguished participants, ladies and gentlemen, we are now moving to the final agenda of this afternoon, the closing ceremony of the Third Aquatic Science International Conference 2025.
To begin this session, we are pleased to announce the best presenter from the parallel presentation session held today. The names will display it on your screen.
Satria from Universitas. Congratulations to all the awardees. May these achievements serve as an opportunity to continue contributing and advancing the field of fisheries and marine research.
Ladies and gentlemen, we now invite the Dean of Faculty of Fisheries and Marine Science, Faculty, Universitas Brawijaya, Professor Absep Aldin Trihanto, to deliver the closing remark. To Professor Asep, screen is yours.
Distinguished keynote speakers, respected participants, colleagues, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon and warm greeting to all of you. Over the past 2 days, we have engaged in the meaningful discussion and engaged innovative idea under the theme of the pollution, protection, and progress, navigating water sustainability in a changing world. On behalf of the Faculty of Fisheries and Marine Science, Universitas Brawijaya, I would like to express my deepest gratitude to our distinguished keynote speaker who have shared their expertise insight with us. The first, Professor Soda Satoshi from Ritsumeikan University, Japan, Professor Dr. Joanna Mankiewicz-Boszek from the Polish Academy of Science, Poland, Dr. Eminier Muzalina Mustafa from University Malaysia Terengganu, Malaysia, Associate Professor Dr. Amiten Ye Hui from University of Malaya, Malaysia, Dr. Umi Yakiah from Universitas Brawijaya, and the last, Dr. Mohan from Beijing Genome Institute, China. I would like to thank all of the committee for a successful third ASIC 2025, to all of the participants from Indonesia and from around the world. All of your contributions have truly enriched the spirit of ASIC 2025 and inspired future collaboration across a border. With all of those proud and achievements, I would like to declare that the third Aquatic Science International Conference 2025 officially closed, and see you again in the fourth Aquatic Science International Conference in 2027. Thank you very much for all of your contribution. Wassalamualaikum warahmatullahi wabarakatuh.
Aquatic Science International Conference 2025.
It has been an honor for us to host this event and to witness the invaluable knowledge shared over the past 2 days. We would like to take this moment to extend our deepest gratitude to all organizers, volunteers, and participants who have worked tirelessly to ensure the success of this conference. Your dedication and commitment have made this event possible, and we sincerely thank you for your invaluable contributions.
We would also like to extend our deepest gratitude to all speakers, moderators, and participants for their active engagement throughout this event. We sincerely hope that the knowledge and ideas shared today will not end here, but will continue to grow and develop in our future endeavors. Let us carry forward the inspiration and motivation from this conference to create a meaningful impact within our communities and beyond. As human beings, we realize that we are not invincible from mistakes. I am Dominic Antoine Erika Putri Sianipar.
And I'm Muhammad Gavin Ahlan Maribowo.
As the Master of Ceremony, would like to apologize for all the mistakes while hosting today's event. Thank you once again for joining us in the 3rd Aquatic Science International Conference 2025.
Under the theme, Pollution, Protection, and Progress, Navigating Water Sustainability in a Changing World. We hope you have a good day ahead, stay healthy, and see you in the next conference. Wassalamualaikum warahmatullahi wabarakatuh.